Hi, everybody. Thank you for joining us today for our webinar, Advanced Multiplexing and Spatial Mapping of RNA Targets using the RNAscope HiPlex V2 Assay. My name is Luke. I'm the webinar manager here at Bio-Techne, and I'm thrilled to welcome you to today's ACD support webinar. I want to thank everybody for joining us today, and I'm excited to welcome our featured speaker today, Dr. Thomas Murphy. Today, we'll start out with a short introduction, and then we have a talk from Thomas, followed by a live Q&A at the end of the webinar to answer any questions that you have. Advanced Cell Diagnostics, or ACD, is part of Bio-Techne. Bio-Techne brings together legendary life science research brands to provide the scientific research community with a comprehensive and world-class product portfolio of reagents, media, extracellular matrices, small molecules, assays, instruments, and more. To learn more about ACD, you can visit www.acdbio.com. And for more info on Bio-Techne, please visit bio-techne.com. And now I have the pleasure of introducing our speaker for today, Dr. Thomas R. Murphy. Thomas joined ACD as a technical support scientist in early 2022 after completing his postdoctoral work and a PhD in neuroscience at the University of California, Riverside. Thomas's doctoral and early postdoc studies focused on the role of astrocytes in seizures, utilizing in vitro and in vivo electrophysiology and fluorescent imaging to study cell volume changes associated with neuronal hyperexcitability. After transitioning to a new project and a new lab, he spent the latter half of his postdoc time using similar techniques to examine the functional role of the synaptic adhesion molecule latrophilin-2 in hippocampal circuits. And with that, we will turn things over to Thomas. Take it away.
Thank you very much for that introduction, and thank you everyone for joining our webinar today. In today's webinar, we will be taking a deep dive into the RNAscope HiPlex V2 Assay. For those joining us who are new to RNAscope, I will begin with a brief introduction to RNAscope technology and assay products. Next, I will focus in on the RNAscope HiPlex V2 Assay, starting with an overview of the assay's capabilities and unique workflow. I will also provide several examples demonstrating how the HiPlex assay can be used, or is currently being used, to help address critical questions across numerous fields of biology. Following this, I will use the remainder of my time to provide an in-depth look at the HiPlex V2 workflow, including a variety of tips, tricks, and troubleshooting recommendations for the assay. This section will also include recommendations for fluorescent imaging, image processing, and data analysis. RNAscope is a powerful in situ hybridization technology which utilizes a unique probe design and signal amplification system to detect nearly any RNA target with unparalleled sensitivity and specificity. It is a highly useful tool for studying gene expression in complex tissues while maintaining spatial and morphological contexts. RNAscope enables researchers to obtain transcriptomic and spatial mapping information for multiple targets at a time in a variety of tissues, using either fluorescent or chromogenic readouts, while also maintaining single molecule and single cell resolution. In short, it is a highly versatile tool for molecular biology research. Because of its versatility and specificity, RNAscope technology has been utilized for a wide variety of applications, from simple expression analyses to identifying cellular sources of secreted factors to validating high-throughput RNA sequencing data. It has played crucial roles in thousands of studies across major fields of biology and biomedical research. This table provides a brief summary of our available assay products. Assay types are generally categorized by target size. RNAscope assays, including the HiPlex V2 assay, are used to detect targets larger than 300 nucleotides and are ideal for standard RNA targets. Basescope assays can amplify much smaller sequences, from 50 to 300 nucleotides in length, allowing for detection of specific splice variants or differentiation of highly homologous targets. The microRNAscope assay can detect targets between 17 and 50 nucleotides long, including microRNAs, antisense oligonucleotides, and other very small RNA targets. Finally, the DNAscope assay is designed for detection of genomic DNA sequences over 3 kilobases in length. Please note that episomal DNA can be detected by RNAscope, Basescope, and microRNAscope assays. For more information about our assay products, please visit acdbio.com/products. The key innovation responsible for the high sensitivity and specificity of RNAscope technology is our unique double Z probe design. Unlike traditional in situ hybridization probes, RNAscope probes comprise a pool of small, Z-shaped oligonucleotides which bind in pairs to the target sequence. Each of these Zs consists of a 25 base region at the bottom, which binds the target sequence, a region at the top, which binds the preamplifier for the amplification tree, and a linker region connecting the two. When both Zs in a pair bind to a target sequence, the preamplifier can then hybridize to the top sequence of the pair. This preamplifier in turn allows numerous amplifiers to hybridize, forming a tree-like structure atop the ZZ pair, which can then be detected by fluorophores or chromogenic enzymes. This amplification strategy ensures highly sensitive detection of RNA targets. Both Zs in a pair must bind to a sequence in the correct order to support the preamplifier and subsequent amplification tree. Since this is extremely unlikely to happen by chance, off-target labeling is virtually non-existent. This lends RNAscope its characteristically high specificity. All RNAscope assays are based on the aforementioned double Z probe and amplification technology and follow the same general protocol. First, tissues are permeabilized using ACD's pretreatment reagents to allow access to the RNA targets. Target-specific double Z probes are hybridized, followed by a series of amplification steps, and labeled by fluorophores or chromogenic enzymes depending on the assay. Because of the greatly amplified signal, individual RNA molecules can be visualized under a bright field or fluorescence microscope, again depending on the assay. This allows for targets to be easily quantified at the single cell level. With that, I would like to now focus on the RNAscope HiPlex V2 assay. The RNAscope HiPlex V2 assay is an advanced, highly multiplexed RNAscope fluorescent assay. Utilizing a unique iterative detection technique, HiPlex enables researchers to detect up to 12 distinct targets in the same tissue section with the unmatched sensitivity and specificity of an RNAscope assay. With the ancillary HiPlex Up reagent, the HiPlex assay can be used for detection of up to 48 separate targets. The ability to detect dozens of RNA biomarkers in a single sample makes the RNAscope HiPlex assay an ideal choice for high-throughput applications and highly granular expression profiling. The HiPlex workflow begins similarly to other RNAscope assays. Following standard tissue pretreatment, probes are hybridized to all targets simultaneously and provide a base for signal amplification trees to be built. Fluorescent labels are then hybridized to the first 3 to 4 target probes, depending on which version of the HiPlex kit you are using. Finally, the labeled probes are imaged using a fluorescent microscope. Those of you who have used other ACD assays will find these steps very familiar so far. Uniquely, however, the HiPlex fluorophores are cleavable and can be easily stripped from the amplification trees. This clears the way for the next set of probes to be labeled using the same 3 to 4 fluorophores, and the sample can be imaged again. This process of cleaving, labeling, and imaging repeats until all probes have been imaged, at which point the images from each round can be registered and analyzed. Due to its unique multi-round design, the HiPlex assay utilizes only a single set of 3 to 4 fluorophores, which are compatible with commonly used filter sets, and requires no specialized imaging equipment or unmixing software to use. The HiPlex Up reagent allows the use of up to 48 probes. Applying HiPlex Up at the end of a standard HiPlex assay will fully strip the amplification trees and target probes from the first 12 targets. This allows the HiPlex assay to be performed again with a set of 12 new probes. Up to 4 cycles of HiPlex can be run using the HiPlex Up reagent. Now that you are familiar with the basic assay workflow, let's have a brief look at some of the powerful ways the RNAscope HiPlex V2 assay can support your research. Because it allows for simultaneous detection of many targets with high resolution, the RNAscope HiPlex V2 assay excels in profiling gene expression in complex tissues. This allows for highly granular analysis of tissue heterogeneity and can be essential for uncovering critical disease pathways or untangling complex cell-to-cell interactions. For example, here is a sample of ovarian cancer tissue stained using the RNAscope HiPlex V2 assay. In a cancer study, there may be a number of immunological markers you want to detect, such as markers of infiltrating T cells or macrophages. With 12 detection channels available in the HiPlex assay, these markers can be stained alongside relevant oncological markers to provide a comprehensive picture of the tumor microenvironment. The HiPlex assay can be an invaluable tool for studying any tissue with complex expression patterns, such as brain tissue. In this example, HiPlex has been used to label mouse hypothalamus with a mix of housekeeping and neuron-specific genes, revealing multiple transcriptionally distinct subpopulations. With the expanded plexing provided by HiPlex Up, multiple separate subregions can be targeted with entirely unique probe sets to identify transcriptionally distinct subpopulations across the tissue. Since spatial mapping is essential for validating single-cell RNA sequencing data, HiPlex also provides an ideal solution for single-cell RNA-seq workflows. The ability to label up to 48 targets ensures that highly heterogeneous cell populations can be easily differentiated by expression profile, and the HiPlex assay has played a crucial role in numerous single-cell transcriptomic studies. In this study, researchers from the Stuber lab at the University of Washington employed the HiPlex assay to help identify specific cell populations in the habenula complex of the brain. The habenula is essential for regulating behavioral states, and in mammals consists of two molecularly and functionally distinct subregions, lateral and medial. However, the expression profiles and cell types within these regions have not been comprehensively defined. Using single-cell RNA sequencing, Hashikawa and colleagues identified numerous genes selectively enriched in either lateral or medial habenula, which in turn revealed several transcriptionally distinct cell types defined by their specific expression profiles. With the RNAscope HiPlex assay, they were then able to directly visualize the spatial distribution of these transcriptionally defined cell types in situ. They further employed HiPlex to confirm and visualize cell populations activated after an aversive foot shock stimulus, helping to identify specific cell types for future studies. Projection neurons carrying pain and itch information are thought to be molecularly and functionally heterogeneous, but until recently the extent of this heterogeneity was unclear. Researchers at the University of California, San Francisco used a retro-trap technique followed by RNA sequencing in mouse LPB projecting spinal and trigeminal neurons to identify projection neuron-specific markers. To determine which neurons responded to each type of stimulus, they used FOS and TRAP2-induced TD tomato expression to label neurons activated by pain or itch stimuli respectively. Using the HiPlex assay to probe for both of these functional markers, as well as the projection neuron-specific markers identified by RNA-seq, Werkberger and colleagues identified multiple molecularly and functionally distinct cell populations among the projection neuron population. Now that you're familiar with the HiPlex V2 assay design and a few of its applications, let's take a closer look at the assay workflow. To get started with the HiPlex V2 assay, you will need the following materials from ACD: the HiBZ oven, humidifying tray, slide rack, and wash tray, pretreatment reagents, wash buffer reagents, the HiPlex V2 detection kit, control and target probes, HiPlex cleaving stock solution, and the image hydrophobic pen. We also recommend at least one set of ACD control cell pellet slides. These can be a simple and effective way to learn the assay protocol without wasting precious tissue samples. In addition, you will also need the following reagents and equipment. Reagents, such as 10% NBF, ethanol, xylene, and PBS, should be prepared fresh for each assay run. 4x and 5x saline sodium citrate buffer, or SSC, can be prepared in advance from 20x SSC stock and stored at room temperature for up to 2 months before use. On the equipment side, please note the Superfrost Plus slides are required to prevent tissue detachment during the assay. Additionally, if you plan to use fixed tissues, you will need either a vegetable steamer or a hot plate and beaker to perform target retrieval. For optimal results, we recommend using a steamer. The HiPlex V2 assay is compatible with FFPE, fixed frozen, and fresh frozen sample types, each of which has specific guidelines for sample preparation, storage, and sectioning. For FFPE tissue, we recommend fixing samples in 10% NBF for 16 to 32 hours at room temperature before embedding in paraffin. FFPE sample blocks can subsequently be stored up to 1 year at room temperature or long-term at 4 degrees Celsius and must be kept in an airtight container with desiccant. FFPE sections should be cut at 4 to 6 microns thick and can be stored up to 3 months at room temperature with desiccant. For fixed frozen tissue, we recommend fixing tissue for 24 hours at 4 degrees Celsius with either 4% PFA or 10% formalin before cryoprotecting and freezing at -80 degrees Celsius. Sectioning should be performed at 7 to 15 microns. Finally, fresh frozen tissue should be immediately snap frozen upon harvest using either dry ice or liquid nitrogen and sectioned at 10 to 20 microns thick. Both fixed frozen and fresh frozen tissues can be stored at -80°C for up to 3 months in an airtight container. Similarly, each of these tissue types requires a specific series of preparation and pretreatment steps. FFPE slides must be baked and deparaffinized with xylene before being rinsed with ethanol and allowed to dry. This is then followed by a target retrieval step, which breaks fixation-induced cross-linking and exposes the RNA targets, and a protease step, which permeabilizes the tissue sufficiently for probes to hybridize and amplification trees to be built. Fixed frozen preparations also begin with a baking step, but are then post-fixed briefly in 10% NBF or 4% PFA. Samples are then dehydrated with standard ethanol series and allowed to dry. This tissue will also require target retrieval prior to protease treatment. Fresh frozen sample preparation requires a longer fixation time and 2 added PBS wash steps, but the preparation is otherwise similar to fixed frozen. However, fresh frozen tissue does not require target retrieval. Here are some important things to remember when setting up the tissue pretreatment steps. Make sure that the HiBZ oven is turned on and set to 40°C at the beginning of the day. Saturate the humidifying paper with DI water and place it in the humidity control tray. Close the tray and place the tray inside the HiBZ oven. Remove your pretreatment reagents from the refrigerator and place them on the bench so they can come to room temperature. If you are using FFPE or fixed frozen tissues, prepare your target retrieval buffer in either a tissue tech container or beaker, depending on which target retrieval method you're using. Make sure you also turn on the steamer or hot plate to bring the solution to the correct temperature. As mentioned previously, different tissue preparation methods will require different pretreatment conditions. Here, I have provided a brief summary of our recommended standard conditions for tissue pretreatment in FFPE, fixed frozen, and fresh frozen sample types, as well as our cell pellet control slides. In general terms, fixed tissues require target retrieval and protease 3, while fresh tissues require protease 4 and no target retrieval, although please note the specific times and temperatures required for each treatment step and sample type. Certain tissue types may require milder or stronger pretreatments than the ones listed here to achieve optimal performance with the HiPlex V2 assay. A list of different tissue types and associated pretreatment recommendations can be found in the RNAscope HiPlex V2 user manual. You can also reach out to us directly for specific recommendations. Once the tissue pretreatment steps are complete, you are ready to run the HiPlex V2 assay. To prepare for the assay, make sure that the HiBZ oven is on and set to 40°C with the humidity control tray prepared and placed inside as before. You will also want to prepare your PBST, 1x wash buffer, and 4x SSC solutions. If you are planning to use the optional breakpoint after hybridization, you will also need 5x SSC. Please note that the wash buffer and SSC solutions can be prepared up to 1 month in advance and stored at room temperature. Finally, you will need to bring your HiPlex amps 1 through 3 and round 1 detection reagents to room temperature, and warm your target probes, controls, and probe diluent to 40°C before allowing them to cool to room temperature as well. As with any RNAscope assay, probe hybridization is the first step of the HiPlex assay. All probes are hybridized to the tissue simultaneously for 2 hours at 40°C in the HiBZ oven, followed by 2 washes in wash buffer, after which the amplification trees can be built. If necessary, you may pause the assay after probe hybridization by storing the slides in 5x SSC buffer overnight, and proceed with the assay the following day starting with the aforementioned wash steps. This will not affect your results. When preparing for probe hybridization, target probes should be pooled and diluted 1 to 50 in probe diluent. Mixed probes can be stored at 2 to 8°C for up to 6 months. Standard HiPlex positive and negative control probes, which arrive in dropper bottles like the one on the right, are already pooled and diluted and can be used directly from the bottle. Here, I have listed the specific genes targeted by our positive control sets, as well as the fluorophores used to visualize them in the 3-round and 4-round versions of the HiPlex assay. These genes have been selected to cover a range of expression levels to assist with assay optimization. As with any RNAscope assay, we recommend that new HiPlex V2 users perform 2 sets of controls prior to running their target probes. First, a set of positive and negative controls in ACD control cell pellets. This serves as a technique check to verify that the assay itself is working as expected, and is a good way to become familiar with the workflow. If there are no problems with the cell pellets, the next set of control probes can be run on the experimental tissue samples. This will serve as an RNA quality check and can reveal any suboptimal fixation or tissue pretreatment conditions which may impair RNAscope results. Optimizing the assay for your experimental tissue at this stage will help ensure high-quality, repeatable results when you run your target probes. Please note that we recommend continuing to run positive and negative controls alongside your target probes even after the assay has been optimized, as controls are essential for setting imaging parameters in fluorescent RNAscope assays. I will touch on this more later in the talk. With that, let's return to the HiPlex V2 assay workflow itself. After probe hybridization and the optional stopping point, it is time to build the amplification trees. This consists of 3 sequential hybridization steps which collectively build the amplification trees for all 12 targets at once. Once the amplification trees are built, the first round of fluorophores can be hybridized. As mentioned earlier, there are 2 versions of the HiPlex V2 assay available. The example here demonstrates the 4-fluorophore 3-round version, which labels 4 probes per round using Alexa Fluor 488, Dilight 550, Dilight 650, and Alexa Fluor 750 fluorophores. These can be detected with FITSI, Sci3, Sci5, and Sci7 filter sets respectively. For researchers without access to a Sci7 filter set, the 3-fluorophore 4-round version of the assay omits the Alexa 750 fluorophore. The assays are otherwise identical, so for simplicity I will focus on the 4-fluorophore version for the remainder of this talk. With the first 4 probes labeled, it is now time to capture the first round of images. To prepare for imaging, slides are counterstained for 30 seconds using the DAPI included in the detection kit, and then mounted using Prolonged Gold anti-fade mountant. We strongly recommend using Prolonged Gold for mounting, as it provides the best preservation of fluorescent RNAscope signals. Finally, these first 4 probes can be imaged. To ensure optimal images for later RNAscope analysis, we recommend always capturing images with a 40x objective or better and using control slides to adjust imaging parameters for each channel. The positive control slide can be used to identify a true RNAscope signal and increase your exposure, light intensity, gain, or other settings as necessary until the puncta in each channel are bright and easily distinguished. Then, starting with these settings, switch to your negative control slide and reduce the same imaging parameters until any background fluorescence is eliminated. These steps will help ensure optimal settings for capturing your target probe signals. With imaging complete, round 1 of the assay is now finished. The next round of the assay will begin with removal of the cover slips and cleavage of the fluorophores. If necessary, slides can be stored in the dark at 2 to 8 degrees Celsius for up to 3 days between imaging and cover slip removal steps. However, it is best to proceed with the next round as soon as possible after imaging, as the mountant will continue to harden during storage and make the cover slip increasingly difficult to remove. Prolonged storage may also increase autofluorescence, impairing signal in subsequent rounds. Cover slips can be removed by soaking the slides in 4x SSC solution for a minimum of 30 minutes and should slide off easily once the mountant is sufficiently softened. If the mountant has hardened significantly, a longer SSC incubation may be required. We do not recommend soaking in SSC for longer than 24 hours. Once the cover slips have been removed, round 1 fluorophores can be cleaved using 2 applications of 10% HiPlex cleaving solution. Cleaving solution will oxidize quickly and lose efficacy if stored, so it is essential to prepare fresh cleaving solution using a new ampule for each round of cleaving. With the round 1 fluorophores cleaved, round 2 fluorophores can be hybridized to probes T5 through T8. The slide is then remounted and imaged once again. Round 2 fluorophore cleavage now proceeds in the same manner as before, with a 30-minute soak in 4x SSC, followed by 2 applications of freshly prepared cleaving solution, allowing for round 3 probes to be labeled and imaged. After the final round of probe imaging, the last set of fluorophores is cleaved and a new cover slip is mounted. Keeping the same imaging parameters used in the last round of imaging, you can now capture a blank image for each fluorescence channel to be used for background subtraction later. This last imaging step is optional, but is strongly recommended to ensure optimal background subtraction during image processing, especially if you are working with highly autofluorescent tissues. To help control tissue autofluorescence, the HiPlex V2 detection kit includes an autofluorescence quenching FFPE reagent. It is highly effective in eliminating fixation-induced autofluorescence, which is most commonly observed in FFPE tissue. However, it can also reduce autofluorescence from other sources such as lipofuscin and red blood cells. We strongly recommend using FFPE reagent when working with FFPE tissue or with any tissue prone to autofluorescence. For tissues with minimal autofluorescence, FFPE reagent is optional. FFPE reagent should be applied just before developing the first round of fluorophores and should be prepared fresh in 4x SSC buffer. We recommend starting with 2.5% FFPE reagent and increasing it up to a maximum of 5% if autofluorescence is severe. If autofluorescence begins to reappear in later rounds, FFPE reagent can be reapplied before the fluorophore hybridization step for each round. When applied as recommended, FFPE reagent will have no negative impact on your RNAscope signal. Before we move on, I would like to provide a few additional tips for performing the HiPlex V2 assay. With regard to the FFPE reagent, you may find that it turns dark or forms precipitates over time after applying it to your tissue. This is normal and will not reduce its quenching ability. When preparing FFPE reagent, we recommend starting with 2.5% and using the lowest concentration possible to quench autofluorescence in your sample. Please note that excessively high concentrations of FFPE reagent may negatively impact your RNAscope signal. When imaging, please try to capture DABI signals with similar intensity each round, as these will be important for image registration later. If the DABI signal dims over successive rounds and visualization becomes difficult, you can reapply DABI again each round prior to cover slip mounting. Last, it is important to note that imaging parameters for each round and channel must be independently optimized, as background fluorescence and signal intensity may vary from round to round. Lastly, I would like to briefly go over the workflow for using the HiPlex Up reagent. As discussed earlier in the talk, the HiPlex Up reagent allows up to 3 additional cycles of HiPlex to be run sequentially on the same tissue sample for a total of 48 markers in a single tissue section. HiPlex Up is applied immediately after the final cleaving step in the HiPlex assay workflow, replacing the mounting and blank image capture steps, which would normally constitute the end of the HiPlex assay. 3 brief applications of HiPlex Up reagent are sufficient to fully strip the HiPlex trees and probes from the first cycle of HiPlex. Please note that the HiPlex Up reagent is not compatible with FFPE reagent. With the trees and probes from the first HiPlex cycle removed, a new set of up to 12 probes can now be hybridized to their respective targets, and the next cycle of HiPlex can be run. Once you have completed the final cycle of HiPlex, we recommend capturing a blank image set using the same imaging parameters used for your last imaging round. These blank images will be used for background subtraction across all prior rounds and cycles of HiPlex. Before moving on to the next section, I would like to briefly touch on a couple of troubleshooting issues. One issue you may encounter is tissue detachment, where samples either partially or fully detach from the slide during the assay. If you see this in your own tissue samples, there are several modifications you can make to your pretreatment protocol, which will help eliminate the issue. They are broken down according to tissue type in the table on the right. Baking steps can be performed using either an air circulating dry oven or the HiBZ oven in bake mode. Before trying any of these modifications, please also make sure that you are using Superfrost Plus slides and that your sections are the recommended thickness for your tissue type. If you are using FFPE or fixed frozen tissue, it will also be important to verify that your target retrieval solution is maintaining a mild boil at 98 to 102 degrees Celsius. And finally, when removing your cover slip for the cleaving steps during the HiPlex assay, make sure that you are soaking the slides until the cover slips fall off easily to avoid tissue damage. One of the most common issues contributing to poor RNAscope signal is suboptimal pretreatment conditions. Overdigestion can generally be diagnosed by indistinct or smeared nuclei. In fluorescent assays, such as HiPlex V2, it can also be inferred by the appearance of autofluorescence in unexpected tissues or excessive autofluorescence in tissues already prone to it. Overdigestion can be treated by reducing pretreatment strength, typically starting with protease time. Underdigestion, which typically results in well-defined nuclei but weak or absent RNAscope signal, can be treated by increasing pretreatment strength. We recommend always starting with the recommended pretreatment conditions for your tissue type. For most tissue types, these conditions can be found in Appendix C of the RNAscope HiPlex V2 user manual. For further guidance, please contact technical support. Finally, I would like to briefly discuss the image processing and analysis options available for the HiPlex assay. Before you can analyze your HiPlex images, they must first be properly registered. ACD has developed a simple but powerful HiPlex image registration software package specially designed for this purpose. In this example, you can see an overlay of 3 image sets with significant misalignment. R1, R2, and R3 are indicating the same cell imaged in rounds 1, 2, and 3 respectively. The HiPlex image registration software can be used to align image sets like this by first registering the DAPI channels from each round, then applying the resulting transformations to the probe channels in each round. This aligned image can then be exported for analysis in standard image analysis software packages. Please note that images must have at least 70% overlap between rounds to achieve proper registration. In addition to its primary function, the HiPlex image registration software also includes 2 powerful background subtraction tools. Background subtraction without a blank image is a one-click subtraction algorithm requiring no additional input and generally produces high-quality results for images with mild to moderate autofluorescence. However, for optimal results, we always recommend background subtraction with a blank image. In this example, RNAscope signal is largely obscured by severe autofluorescence, visible in the blank image to the right. Using background subtraction without a blank image produces acceptable results, but fails to eliminate several bright artifacts as indicated by the yellow arrows. These artifacts may be incorrectly detected as RNAscope puncta during later image analysis. By contrast, blank image subtraction with the blank image effectively removes these artifacts, and the resulting image accurately reflects the RNAscope puncta visible in the original data. When analyzing your RNAscope images, the important measures to consider are the number of individual puncta and the number of clusters. Each single punctum represents a single RNA molecule, while clusters of puncta represent overlapping signals from individual puncta. Variations in puncta size or intensity, as highlighted by the yellow arrows on the right, arise primarily due to differences in the number of ZZ probes bound to a target RNA molecule and do not represent any functional difference in expression. There are multiple ways to analyze your RNAscope data. One recommended approach is to assign semi-quantitative scores based on the average number of spots per cell and the number of clusters. This is a quick way to analyze targets with relatively homogeneous expression across a cell population and can provide a helpful starting point for more complex analyses. It is also a useful metric for evaluating assay performance with control probes. Our standard negative control, DAP-B, is expected to score 0 in all tissues, while our standard positive control, PPIB, is expected to be approximately 2 to 3 in most tissues. This is only one of several analysis approaches you might consider. For more information on RNAscope analysis methods, please see our data analysis guide. RNAscope HiPlex data analysis can be performed using any suitable image analysis software. We recommend using Fiji or ImageJ, QPath, or Cell Profiler, all of which are free and relatively simple to use. A technical note and recorded webinar detailing the RNAscope analysis workflow for each of these software packages can be found on our website by visiting the links at the bottom. You can also reach out directly to the technical support team for assistance with your analysis. To briefly summarize, the RNAscope HiPlex V2 assay is a powerful expansion of our existing RNAscope technology, enabling detection of up to 48 targets in a single tissue section with single cell and single molecule resolution. Due to its unique iterative detection system, consisting of multiple rounds of fluorophore hybridization, imaging, and cleaving, the HiPlex assay is able to utilize a small group of fluorophores compatible with standard fluorescence filter sets and requires no specialized microscopy equipment or on-mixing software to use. With the ability to detect so many targets at once, HiPlex allows researchers to profile complex tissues and map spatial distributions for dozens of genes at a time, and is an excellent complement to high-throughput workflows like RNA-seq. This makes it an ideal tool for high-impact studies in cancer, immunology, neuroscience, and a variety of other fields. Finally, the HiPlex image registration software supports the assay by providing a simple but powerful tool for registering HiPlex images and subtracting background fluorescence. Before concluding, I would like to briefly highlight the additional recorded webinars available on our website, which contain a wealth of information about our other assay products. For pricing, ordering, and other sales inquiries, please reach out to our sales team by email at acd_sales@bio-techne.com. For technical inquiries, troubleshooting, and other assay support issues, please reach out to the technical support team. You can reach us by phone at 1-877-576-3636, by email at support.acd@bio-techne.com, or by using the online chat feature available on our website. Thank you all very much for joining us today, and I'll be happy to answer any questions you have.
All right, fantastic. At this point, we'll welcome Thomas back. Thomas, thank you for that presentation, and welcome for the Q&A here.
Thank you.
Fantastic. All right, let's get right into it with some of these questions here. Question number one, can I perform the IF/IHC co-detection with the HiPlex assay?
So for IF/IHC co-detection, we haven't validated the HiPlex assay with protein co-detection. Some customers have successfully done sequential immunofluorescence with the RNAscope HiPlex assay, meaning performing immunofluorescence or IHC immediately after the assay. But the only precaution with that is that the only thing to note about that is that the HiPlex assay does not include a peroxidase quenching step. So if you want to perform that, you need to be careful not to use HRP conjugated antibodies, because that could result in very high background.
Gotcha, thank you. Should the FFPE reagent be used only on FFPE tissues?
No, so the FFPE reagent was designed for FFPE tissues primarily because FFPE tissues tend to be the ones with high autofluorescence. It was designed to combat fixation-induced autofluorescence, but it can help combat autofluorescence from several different sources. I mentioned lipofuscin and red blood cells in the talk as a couple of examples, and essentially we're recommended for any highly autofluorescent tissue. By the same token, not all FFPE tissues will necessarily need it. So it's best we recommend it generally for FFPE tissue, but it's not necessarily a requirement. It's just recommended for highly autofluorescent tissues.
Gotcha, and one more question on FFPE. Can you use that reagent with the HiPlex Up assay?
No, it can't be used with the HiPlex Up assay.
Gotcha. Why does the HiPlex V2 FFPE pretreatment require protease 3 instead of protease plus?
So different RNAscope assays require different levels of pretreatment, different digestion, basically. And for the HiPlex assay, we've found that protease 3 works better for allowing at least for FFPE tissue, for allowing the amplification trees to be built.
Gotcha, thank you. How many targets can you analyze in FFPE tissue sections?
So again, this kind of gets back to the question of FFPE reagent versus not. So if you're using FFPE reagent, you're limited to 12 targets because the FFPE reagent is not compatible with HiPlex Up reagent. If your FFPE tissue or your tissue in general does not require FFPE reagent, then you can apply the HiPlex Up reagent and expand your assay up to 48 targets.
Fantastic. Can the positive control probes and target probes have a crosstalk issue when they are run together?
No, they shouldn't. So the way there may have been some miscommunication there in the talk. When you're running them together, they're run on separate slides. So they're going to be they're targeting separate, obviously targeting separate genes, and they're run on separate slide. So there's not going to be any issues with crosstalk. You're basically going to have a positive control slide, a negative control slide, and your target probes on a separate slide.
Gotcha. Can you purchase that FFPE reagent separately, or is it just in the kit?
No, the FFPE reagent is only with the HiPlex kit. That's part of the HiPlex kit.
All right. Are blank images recommended to be taken after all rounds are completed, or should blank images be taken after each round?
We recommend only capturing the blank images at the end of the last round, whether that's at the end of the single HiPlex assay or at the end of the multiple cycle HiPlex up expanded version. Whatever happens to be the last round of HiPlex you're running.
Gotcha. All right, tons of questions coming in the chat. Thank you so much. We will continue taking these until we run out of time, but keep on typing them in that question box on the right side of your screen there. Does it have to be specific fluorophores or one, can you label fluorophores of choice? Say some fluors are brighter and or imaging equipment is available that could be limited to a particular set of filters.
So the fluorophores are preset. There's the two different, I mentioned there's the two different assay types, one of which has the three fluorophores, the Alexa fluor 488, Dialite 550, and Dialite 650, and the second one which has those first three plus Alexa 750. So you're limited to those channels, but you do have some control over which genes you actually assign to those. So when you're setting up your probes initially, when you're ordering your probes initially, you can set them up so that we generally recommend this actually, setting them up so that you're ordering your probes in order of from 1 to 12 in order of expression level. So from lowest expression to highest expression, and then within each group of 3 or 4, depending on the assay, from highest to lowest expression. So your account manager can explain that a little bit better than I can by audio. But the basic reason, basically what you can do there is you can assign your probes to whichever specific channel within each round of HiPlex that you want to. We always recommend that particular order I mentioned because the green channel, if there's going to be autofluorescence, the green channel is where you're going to see it. So you want your highest expressors to be on the green channel so that they can, so that this signal to noise is highest.
Fantastic, thank you. Can the cleaving step before HiPlex up reagent be omitted?
That is a good question, actually. I'm not certain.
No worries. We will get back by email to the person that asked that.
Yeah, of course.
Question.
Reach out to us by email and we'll be happy to look into that further.
Fantastic. So how long does this whole process take from start to finish? And they've given a particular example here. I run the V1 12 flex, 4 rounds of 3 fluorophores. So how long does it take to run this HiPlex V2?
So it can kind of vary depending on how much you're willing to do in a day. I think it's usually going to be at least a couple of days. And I had this written down. I calculated this out at one point because I got this question from a customer once, but I can't remember off the top of my head. I think I was calculating it out based on the longest possible amount. The longest possible amount, I think, or longest possible time is upwards of a week. But that's if you're using, that's if you're going with the maximum amount of time that we recommend letting the slides sit before you move on to the next round. If you're keeping it to just let's say 1 round per day or a couple of rounds per day, you could have it finished within a few days.
Fantastic, thank you. still a lot of questions coming in and still time to add more. So again, you can use that chat box on the right side of your screen to send those in. Any special recommendations for using non-adherent cells, PBMCs as a starting point?
So we haven't validated that application, and we don't support it. Some users, I think, have used cultured cells with HiPlex. We haven't, in our experience, cultured cells have a risk of detaching from the slide over time because of the multiple rounds of washes and reagent applications. So it's generally not recommended to use cultured cells with HiPlex.
Gotcha. Another question here, when can we break overnight? Each round of imaging takes 4 to 6 hours or so, going from the start of a 12-plex to final imaging. The person asking this is a solo lab tech wondering when to break up the work steps of this assay.
So the breakpoint would probably be after imaging. The slides can be stored for up to 3 days before moving on to the next round. You can also, depending on how hardened the mountant is, you can also potentially soak the slides for. You may need to soak the slides for up to 24 hours, but it's recommended to do that as short a time as possible. So that's probably not the best breakpoint. Usually the best breakpoint is to just keep is the storage point right after imaging.
Awesome, thank you. A question, can this assay be run on the Ventana platform?
No, as of now, we don't have an automated version of the HiPlex assay.
Gotcha. We have a user here. I've been using Opal dyes. Can this be used in the 48-plex?
Opal dyes in the. So Opal dyes are the HiPlex is not HRP based. It's a different chemistry from. It's a completely different chemistry. So it doesn't use Opal dyes or any other TSA type dye.
Gotcha, thank you. We'll give just a couple more seconds here, but it looks like we're coming to the end of our questions that are coming through here. We've been doing a lot of responses in chat, so if we didn't get to your question live here and we didn't get to it in chat, you can expect a follow-up from the support team with an answer coming to your email, the same one that you used to register. But at this point, I wanted to just thank everybody for jumping in today. Thank you, Thomas, for the presentation. Excellent, well done, and thanks to everybody for joining us. We do have a post-event survey, which you'll see after this webinar ends. Would love to hear what you think about the webinar and anything that you'd like to hear about for future topics. So if you have a few minutes, please do take that survey. But as always, on behalf of the team at Bio-Techne and ACD, thank you very much for joining us today, and we hope everybody has a wonderful rest of their day.
Thanks, everyone.